On our previous presentation, we had finished up talking about plasma membrane specializations. So now, presentation, we were talking about plasma membrane specializations. So now, let's move on to the cytoplasm and the organelles that lie inside of it. Now, the cytoplasm is the material that's outside of the nucleus of the cell, but it's inside the plasma membrane. And it is a gelatinous kind of goo substance. It's mostly water, and it contains cell organelles. The cytoplasm has three major elements or components. There's the cytosol, which is the fluid component, and that fluid contains ions and electrolytes and sugars and everything else that the cell needs to function, or that the organelles need to be able to function. Now, the organelles are the metabolic machinery of the cell, and just like small organs, they perform specialized functions for the cell. Now, some cells also have inclusions. For instance, fat cells have a large vacuole that's just full of the triglycerides, full of the fats. In plant cells, there are vacuoles that are full of water. Just to give you some examples. Now, this is sort of a generic cell. There's not a single cell out there that looks like this, but this has all of the components that we're going to discuss. There's the nucleus and the nucleolus, and then we have the endoplasmic reticulum, both the rough endoplasmic reticulum and the smooth. We have mitochondria. We have lysosomes, we have Golgi apparatus, we have centrioles, and all of these serve specific functions. So let's start, and in no particular order, talk about each of these. Now, mitochondria, these are small organelles, similar in size actually to bacteria. And these are sometimes referred to as the powerhouses of the cell, because they have the metabolic machinery to convert the breakdown products of glucose into ATP. Without the mitochondria, we could get, per molecule of glucose, about 2 ATP. With the mitochondria, we can get 34 to 36 ATP. So this allows multi-celled organisms to exist. Without this, we would not have complex life forms. So, powerhouse of the cell, as I said, similar in size to bacteria, which leads to something called the endosymbiotic theory. That theory states that there at one time was a small bacteria that was able to produce ATP that got eaten by a larger microorganism. And instead of being destroyed once it was taken in by the larger microorganism, it continued to survive in that microorganism and thrive because it was protected from the outside environment, and the microorganism that had eaten it was getting the benefit of additional ATP. Now, one thing that supports this is that the powerhouses, the mitochondria, replicate independently. They grow in numbers when they are in an environment that supports it. So if they're in an area that has a lot of oxygen and nutrients, then they will replicate. And when the cell undergoes mitosis, then some of the mitochondria go with one cell and some go with the other. They are kind of changing shape all the time, almost like they're swimming or wiggling around. They carry out all of the reactions where oxygen is used to break down food. So every breath that we're taking in is to support the activity of the mitochondria. All of the air that you breathe is working to aid the mitochondria in the breakdown of food products so that we can have energy. Ultimately, they provide ATP, which is the cellular energy for our bodies. It is the exchange, as it were, for all activities. Now, another cell organelle, which all cells have, is all cells have to make proteins. And so all cells have ribosomes. Not all cells actually have mitochondria. All human cells do, but there are microorganisms that don't have mitochondria. Everything, though, has a ribosome. And it's made of proteins and RNA. It's manufactured in the nucleus, in the nucleoli area, and then it migrates out into the cytoplasm, where it becomes a site of protein synthesis. Now, in the cytoplasm, it's found in a couple of different locations. There's free-floating ones in the cytoplasm, and there are those that are attached to the rough endoplasmic reticulum. Now, those that are free-floating in the cytoplasm make proteins specifically for the use of the cell. Those that are part of the rough endoplasmic reticulum make proteins that can be exported or taken out of the cell. So, in terms of maintaining internal structure, those that are free in the cytoplasm are making the proteins necessary for that. On the other hand, if I have a cold and I'm making a lot of mucus, that mucus is being made by proteins or ribosomes that are located on the rough endoplasmic reticulum. Now, we mentioned rough endoplasmic reticulum. There are actually two forms of endoplasmic reticulum. There is rough ER and smooth ER. And just to save time, we'll refer to these as ER. They are both fluid-filled tubes that carry substances and store substances. The rough endoplasmic reticulum looks kind of like sandpaper. It is studded with ribosomes, and it synthesizes and stores proteins. Smooth endoplasmic reticulum doesn't have any protein manufacture. Instead, it has a lot of enzymes on the surface which help it function in lipid metabolism, detoxification of drugs, and pesticides and other substances. And so cells that are detoxifying lots of substances are going to have a higher level of smooth endoplasmic reticulum. For instance, the liver has more smooth endoplasmic reticulum than other cells do. Now, the rough endoplasmic reticulum is involved in protein synthesis. We'll talk a little bit more about DNA and RNA and protein synthesis later, but what we have are the ribosomes, which are reading messenger RNA, which allows it to code for the production of the protein. The protein gets stored here in the rough endoplasmic reticulum until a sufficient quantity is made that can be packed up into a transport vesicle, which then moves the material from one location to another. Now, I think of this kind of being like this is a factory floor. This is a machine that's reading a blueprint or a plan of some sort, making a product that goes to the warehouse, and once there's a sufficient quantity in the warehouse, then it gets packed onto a truck and shipped out elsewhere. And the elsewhere that it gets shipped out to is a structure called the Golgi apparatus. Now, the Golgi apparatus modifies and packages these proteins, and it produces several different types of packages. So this stuff is an incomplete component that gets shipped to here to be modified. I used to work at a factory. I worked at the General Electric plant down in Somerset. It was a headlamp component factory. Now, we didn't make headlamps. We made the reflectors and we made the lenses. So we made these parts. We shipped that to the assembly plant, which is a factory here in Lexington, another GE plant. And they put the reflector and the lens and the light bulb and other materials together to actually make the completed headlamp. So the parts factory is like the endoplasmic reticulum. The assembly plant is like the Golgi apparatus. Once it has assembled it, then that gets to be shipped either outside, let's say this is for export. Some parts of the Golgi apparatus will repair damage to the cell membrane. So every time something gets exported, some of the Golgi apparatus is used to patch holes, basically. And then there's also lysosomes, and these are structures within the cell that the cell uses to break down components it no longer needs, or to break down materials that it has ingested. Now, just so we've got this whole sequence, we are making messenger RNA in the nucleus. The messenger RNA goes out to the rough endoplasmic reticulum, which is actually attached to the nucleus, where the messenger RNA gets read by the ribosomes so that proteins are made and stored in the rough endoplasmic reticulum. Sufficient quantity of proteins then gets put into a transport vesicle, which carries it to the Golgi apparatus. The Golgi apparatus modifies and packages those proteins into a finished product, which either get excreted by exocytosis. You can have portions of the Golgi apparatus just repair, but usually you're going to see that repair occur as a part of a transport vesicle goes out. And the third pathway is that you can have these little vesicles with very strong digestive enzymes that can merge with other vesicles when the cell brings something in to help destroy it. These vesicles also break down cellular components that are no longer functional. Say we had a mitochondrion that has died or is non-functional in some way. These vesicles will attach to it and destroy it. Now, these other organelles, we mentioned lysosomes. These contain enzymes that digest worn out or non-usable material within the cell, or, again, things that the cell has brought in. We also have peroxisomes. Now, peroxisomes, they have something called oxidase. Now, oxidase is an enzyme that can break down peroxide. The peroxisomes can help detoxify harmful substances, again, such as alcohol, formaldehyde. They destroy free radicals. And a free radical is a highly reactive chemical that can do a lot of cellular damage. Peroxisomes also generate independently, just like the mitochondria. What they do is they bring in substances, they make their oxidase, and when they have a sufficient quantity, they just pinch in two, and now we have two of them, and then those two start to absorb and build new materials, and again, pinch in half as needed. So when you pour peroxide on an injury and you see that peroxide foam up, what's happening is that these oxidase enzymes are reacting with the peroxide to release these gases and these materials. Now, the cytoskeleton. This is a network of protein structures. They go throughout the cytoplasm and they provide a framework and transport mechanism for the cell. They help the cell hold its shape, for one thing. Now, I have nerve cells that have to stay in contact with other nerve cells and with muscles, and this internal framework allows it to keep that appropriate shape. I want to talk a little bit more about that in a few minutes, but we are just about out of our 15-minute time frame, so we're going to stop here and we will pick up on an additional discussion. Thank you very much.